Great.
Oh,
Hi, everyone. Hi, Phil. Thanks for the intro. Great. So I think we can get started now, Davide. Hi, everyone, and a really warm welcome to our workshop today. My name's Leena, and I'm joined with my colleague, Davide, to present to you today's workshop. And I'm just going to share just a second. Okay, so hopefully you should all see my screen. Maybe give a quick thumbs up because I can't actually see on my end whether you can see the same thing. Okay, great. Brilliant. Okay, so our workshop today is called The Near Future of Regulatory Submissions, Embracing Reproducibility, Open Source, and Interactivity. So we'll start off the workshop with a bit of an introduction to Davide and myself. This workshop's going to be a bit of a mix. So we'll do a bit of context sharing to begin with. Then we're going to go into some more hands-on programming. But first, let me introduce you to myself and Davide. So Davide, you want to start?
Yeah, just to mention that some of the materials was inspired by another Pharma Workshop and is linked in the presentation. Yeah, and yeah, I'm Davide, and I'm working mainly in the background of making things works. So I'm checking that all the issues on GitHub for our tabulation software and helping the TL team and so on. And I am very eager to get into the future of regulatory submissions, and I matured an idea of what it will look like, and together with Leena, we made this presentation and this workshop to try to grasp that kind of vision. Yeah. Leena?
Yep, thank you. So I have been at Roche now for 5 years. I'm a statistical programmer. Previous to that, I worked in academia where I was actually using R in a lot of my day-to-day work. At the moment, my role is kind of multifaceted at Roche. I work in late phase studies supporting the analysis of data for clinical studies, so producing SCTM, ADAM, tables, listings, and graphs. And I also provide oversight of other programmers and work very closely with them for our deliverables. My passion for programming has always been very strong with R, I would say. I'm an advocate for the use of R and open source packages internally for programming activities. And at Roche, I onboard other statistical programmers as they begin to use the new tooling capabilities and get introduced to new ways of working with R. So like I said, a bit of a multifaceted role. And in addition to this, I'm the product owner for Nest TLG capabilities. And we'll go into a bit more detail about what this looks like and what Nest is overall, and specifically which products kind of fall under the scope for myself and Davide. And being product owner essentially just means that you develop a vision for your products or packages, and manage and prioritize work for the team. And it also means I have a really close relationship with end users as well, so the people who use our packages, and to continuously improve them and improve the vision for the future as well. Okay, so I'll cover a bit of the schedule for today. We have 2.5 hours for the entire workshop. So we'll discuss a bit more of a detailed background to the workshop. So why change? I think we're already a bit over time, so the timings might be a bit off. But yeah, we may have to squeeze bits where we can, Davide, to try and make 2.5 hours really happen. Then we jump into the first part of the hands-on programming, which is this getting there section. We have around 50 minutes allocated for this. We'll take a quick break for about 10 minutes, and then we go into the second part of the hands-on programming task. again for another 50 minutes, and then we will wrap up with some general conclusions, what we expect to see in the future, and then some additional resources for you to go away with. And maybe it's been explained already, but yeah, please add any questions you have to the public Zoom chat. We'll try and address them maybe in the moment or maybe at the end, depending on the context and if we've got time for a Q&A session. But both Davide and I will be looking out at the chat to answer if there are any questions on the fly. Okay, so with that, let's get into some of the context. So let's talk about why change, why we're seeing this shift in dynamic. Many of you already know about industry efforts in modernizing solutions in pharma in a regulatory setting. And really the purpose of this workshop is that we want to highlight this change and discuss how this shift has benefited us as an industry and brought us closer to regulatory submissions in R. So first, let me briefly cover what our main tasks are, which is reporting out clinical trial results. And really the purpose of regulatory submissions is to show efficacy and safety of our new drugs. And what this means for us as statistical programmers or data scientists is to deliver analysis reports, data sets, and software as well. So these are in the form of SDTM, of ADAM, our tables, listings, and figures, but most importantly that we wanted to highlight is our software. And this is a deliverable of our reproducibility of the results that we present to health authorities. So health authorities need to be able to reproduce these key results that we send to them. So with that, why would we migrate to R in clinical trials? And what's the purpose of migration of the tool that we use to deliver in our clinical trial? And so I'm going to quickly talk through some of the benefits that we can see for everyone. Firstly, we see a benefit in cost efficiency. R is free and this reduces software costs. We also see flexibility in our solutions. R offers greater customization and a vast package ecosystem as well. And moving through from my experience from academia into industry, we can see that this customization really flows through the two different areas of academia and industry as well. We also find a sense of community and support for collaboration. We have a really large and active R community, which drives our rapid innovation of our products and our packages. And collaboration in particular sparks new ideas and momentum in our tools. So let's explore this point of community support and collaboration in a bit more detail. So this really comes off of other wonderful initiatives, such as the one that we have today, such as R in Pharma, and also many others. We've left a link to a webinar here where you can kind of get to understand most of the other groups that are supporting R. But also we wanted to mention other initiatives such as our consortium, which has the support of a vast group of companies. Another one worth to mention is Pharmaverse, which is a network of companies who actively contribute to the development of open source packages. And something really key to highlight here is that the number of companies who are actively contributing is just amazing. And you can see all these different companies and people contributing to different working groups and also things from the Pharmaverse, such as Pharmaverse blogs. I think it's really great and it really shows our true community spirit. Okay, so what's in it for us and what's the benefits for individuals or companies in particular? Well, in the past few years, we've seen a shift in our talent and also the complexities of clinical trial design and the way in which we collect data in trials. So this is really talking to why we see this and what are the factors that really can help us harness the power of open source. The first thing, and I already touched on it, is our talent pool. The shift in our talent base already tells us that more and more professionals, as particular graduates, have familiarity with R or other languages such as Python and the use of Git. And this really enables our productivity in the setting of open source, as newer talent that we hire have these experiences to hand. We also gain enhanced transparency, and this is especially important to our health authorities and as developers, as a group of developers, to identify where we can find gaps in our landscape of packages. So R's open source nature really ensures that full transparency and reproducibility can be met. Integration, R easily integrates with modern data tools and platforms, so this really accelerates us in our industry. And R is a transversal skill, so other business units outside of pharmaceutical industry may also benefit from R programmers. And the final point, scalability in complexity. So what does this mean? It essentially means getting the latest developments and adoption of the newer solutions a lot faster and at a lot larger scale. We can see that very novel methods can be readily applied to clinical trials, growing in complexity. So where we have more difficult clinical trials, with the data that we collect, these novel methods easily can be applied to our clinical trials and can just accelerate at a much faster pace. So with all of these, we aim to bring a lot more efficiencies to our end users and to our clinical trials by embracing open source languages, and in the end, develop insights generation at a much faster pace. So now I'll specifically talk from a Roche point of view and talk through our journey as a company and what we have achieved to date in terms of the modernized solutions using R. So the first time point, which is like a landmark moment for us, is 2017, where we established our proof of concepts. So this was the creation, the development of our tables and Teal for interactive work. They were both launched and presented at R in Pharma during this time. And shortly after this, internally, Nest tools were used for exploratory purposes. Following this, established the kind of open source movement. So our tables made our first CRAN release in 2021. And we followed by open sourcing the code base as well to kick off collaboration with other companies. And this really helped shape the external environment outside of just our company. So packages were made readily available openly, and this really increased our external presence. So the next big landmark moment, I would say, is in 2023. So at Roche, we developed an in-house platform which serves as our backbone for data science deliverables in a validated environment. And this was really to deliver our purpose and our vision of submissions in R. Generally, at our organization, we have a mix of SAS and R programmers, all at different and varying levels of experience. And our platform for data scientists really leverages the use of diverse tools, diverse languages in the way that we work. Another point to mention here is really when we started to establish some good practices on the newer ways of working with open source packages, dealing with validation, and getting to grips with a new kind of hybrid way of working, and an entirely different landscape for our submissions as well. And it led us to this really nice moment this year, actually, where we had our first end-to-end R submission at Roche. And we've recently heard from our own colleagues in a really well-attended webinar explaining Roche's end-to-end journey to submission, and about the experience that we all shared on the first R submission. We're really pleased to say that tools such as ours in Nest played a really huge part in this R submission. Not only did our users of the tools learn so much from this experience, but also our development team. So that was a really pivotal moment for us. And it showcased the benefits of all of the efforts that I've mentioned so far. So the community groups that we've established to really help steer this event. So if you'd like to watch the webinar, and I really encourage that you do, we've left a link as well in our slides for you to see as well. And just on a personal note, I really hope that this kind of trajectory on our journey really continues and we bring about some more examples of success. So now I'll hand over to Davide. I'll continue driving the slides for you, Davide.
Thank you, Leena.
Yeah.
So as already someone asked in Zoom, there is what is the feedback from health authorities? So how R is seen by FDA or other health authorities? And we can say that it's increasingly accepted. And I watched a very interesting talk that is coming from last year, and about the FDA and their opinion. In general, Leena,
Yeah.
I mean, while talking about this, there is always the idea of kind of revolution, like changing the paradigm and so on. But after talking with people and so on, it seems that to me that is more an evolution. So it's something that is integrating slowly into into all pipelines. So it's coming more often that all submissions have a part in R, and that integrates the clinical trials reports and results. Next. Also, there are very, strong efforts important efforts to talk with FDA. In particular, I will cover mainly FDA, and as it's one of the main driver at the moment of this innovation. And also because there is this a very interesting talk. It's linked in the slides. Here you can see it, but if you go to the website that I posted at the beginning, you have all information on all the links. And it's an adoption series. So basically, the R consortiums prepared a working group called Submission Working Group that talked with FDA about collaborating and having our submissions in R. In particular, they were planned for pilots, and 3 are finished and successful. Don't dwell on the details, but pilot 1 is mainly R files. So really from scratch, tables, listing, and figures. Then pilot 2 is a Shiny interactive application with RM and package light. Then pilot 3 that was successful, it was finished. This is coming from last year, this slide. And it involves Adam creation and Adam datasets, and is using again R files. The next one, I think, is still happening. The pilot 4, sorry.
Oh, sorry.
Not a slide. Pilot 4 is it's the last step, and I think it's a very important one, and we will see afterwards why. Because it's proposing a self-contained environment, so a container or a WebAssembly, so a place where you have all the packages already installed, and this should, and I believe will, guarantee reproducibility of the code. Next. So this is from pilot 1, and you could see that this is a classic demographic table. Nothing too particular about this, but when I saw this, I said, this looks familiar. I mean, of course, they are very similar, But then I said, okay, let's see pilot 2. Next. And pilot 2 had some interactive Kepler-Meyer plot. And I said, also these filters, these things look also very familiar to me. So I told myself, let's check the code of pilot 2. So Leena. And probably you can see, I think I can annotate that here there is teal. Sorry, horrible annotation. And R tables. And also if you check the code, this is basically R tables, which is our main tabulation software, the one that we started working on since 2017 with the first POCs. And then teal, that is, I mean, you might be aware of it, is our interactive platform. Next. So I was very happy about that. Ooh, I need to kind of remove this. Okay, yes, sorry. So all good, looks amazing, but what are the findings and what are the issues that FDA is facing? So what are the complaints about our submissions? And the first one it's about different versions. So R and open source tools change very fast. And it's a pro because you have a large community that works on it, and you have access to novel methods and so on. But it means that every time to guarantee reproducibility, you need to be very, very sure about the versioning and have exactly a certain snapshot of the environment. It means also for less experienced user that might have problem with also operating systems. So running, for example, pipelines on RStudio on Windows could be a bit different from running it on Linux. And then lastly, and I think it's one of the most important, is that they said that interactive features could be prone to p-hacking. So to, let's say, tailor your data to show what you want. And next. And but I argue that the first of two issues will be solved by Docker and WebAssembly. And other proprietary software do have the same issue. You need to specify the versions of the software you use, and it's the same in this case. Regarding p-hacking, also during the meeting there was someone saying that p-hacking is not technology dependent, so you can use it with anything. And of course, the ease of use and tweaking possibilities of interactive features, it's something that brings us more on the exploratory world, because of course you can always do this at any time. But we will see later between the last parts of the actual coding workshop that it's something that is actually a very powerful tool. For example, for responding to the FDA. So if they say, okay, this happened for this case, but what if you do something else? And in this case, you would have to rerun everything. Instead, with an interactive feature, you just check with some buttons. And I know that the FDA has some exploratory tool on their side, so it's something that is coming more prominent with the time. Next. For in general, as a guidelines, I don't know if someone probably had a question on how do we deal with R-based submissions. For the FDA, they say just tell them, use cron and curate the repositories for packages and minimize dependencies, but on the sponsor developed packages. But all of this will be solved by WebAssembly and containerization, and on the CI/CD. So we will not touch too much of that, but all our packages are continuously tested every day with thousands of tests with unit tests and regression tests and these kind of things. And they are validated on the validator hub that some of our colleagues maintain, which is a bit, I think is more stringent than CRAN. And all our packages are CRAN because it's open source, so yeah, that's also there. And of course, provide the documentation and detailed comments. So we have we put a lot of effort in this, and I think it's very relevant because it makes everything more readable. But of course, also I was talking with some colleagues about readable code, and I mean, if it's open source, it's already a very good pro for readable code. But that's another discussion, so it's for another time. Leena, now to you.
Yeah, thank you. Yeah, so like Davide mentioned, reproducibility and validation. Anyway, our packages are in CRAN and has extensive CI/CD testing, precise documentation and versioning. The open source element also allows for collaboration and transparency and really accelerates our industry towards standardization, especially if we adopt a lot more wider. And this could also potentially lead to faster submissions if we're all kind of following the same trajectory. And finally, interactivity. So Teal, which is our more exploratory tool, integrates standard tables, listings, and figures, and enables you to have novel findings on the fly and a faster reviewer responses. So you wouldn't need to go back and regenerate static TLGs. Really, you'd have this all to hand using interactive elements such as Teal. Okay, so with that, I'll explain a bit more about Nest. So Nest is a collection of open sourced R packages, which enables fast and efficient insights generation under clinical research settings for both exploratory and regulatory purpose. And really our aim within the Nest team of data scientists and experts and our team of developers is to create ready to adopt solutions for TLGs for regulatory purposes and also exploratory elements as well. So this is a showcase of our landscape of Nest products. It's actually a very trimmed down version of our landscape of packages and products. There are many, many more in the Nest ecosystem, but we thought we'd keep it very exclusive to this workshop, especially to the ones that we're going to introduce to you a bit later in the hands-on component. So TLGs can be produced in various ways using different packages, and you'll get to experience that today as well. But what's great about our suite of Nest packages is that our vision for the products is to aim towards a more complete solution for R-based insights delivery. And this entire landscape of tools we have that we have developed enables each of our products and our packages to work together to create a more streamlined process for your deliverables. So if I just introduce them very briefly, we have our set of foundational R packages to support the generation of outputs, and these are R tables and Turn for statistical components. We also have Teal, which supports interactivity and exploratory web apps based on a Shiny framework. And primarily at the moment we use for exploratory purposes and diving into our data on an exploratory level. Next, we have our catalog for common TLGs and apps. So the TLGC is also one that you'll get introduced to today, or the TLG catalog. And this is a very comprehensive catalog of commonly used tables, listings, and figures for the purposes of submission. And in companion for exploratory purposes, we have the Teal Gallery, which showcases ready-to-use customized modules for Shiny apps and ties nicely hand in hand with the TLG catalog. As you can see, many examples of Shiny modules are also available as a static TLG format and vice versa. So this entire landscape of packages and these two products work coherently to produce regulatory outputs and exploratory insights generation, like I mentioned, with the help of teal. Okay, so now we've explained the context and the way in which we're working towards using R for regulatory submissions as an industry with a perspective as well at Roche for our first R submission. We would like to give you the opportunity to learn our suite of tools in Nest to bring you a lot closer to regulatory submissions in R. So this is where we go to our more hands-on portion of the workshop so that you can understand the context behind the development of our tools and how we actually put it to use. Okay, so Davide, shall I now hand to you and we get started? Mm-hmm.
So last chat question. So we are using a Shiny test not since too long, I think, but it allows to do a lot of coverage that we couldn't before. But anyway, it's integrated still with manual testing, and so every release we test all the applications and all the options and so on. In general, they're also aimed tests, so we have like the change log, and we check on the test specifically. We test specifically the new features and so on, besides general random clicking, let's say. Yeah, this was beside the unit testing, like called self maintenance, that was the original coverage for complete applications. We always used to do this. Now we have also Shiny tests, which is quite a good upgrade. Yeah. Leena, you're in charge of the questions and the chat.
Yep.
So this is the preparation slide. So we recommend to use our Studio Cloud, POSIT Cloud. So where there is the workspace for completing the exercises. It is there are not there is no any code that will guide you. So I invite you to use the website and also the exercise on the website to see roughly what is going on. In general, just listen here, and then during the 10 minutes you can it's all about making from scratch or taking information from some source. So this is because this is the way nowadays I think everything will happen. So we will see for the first exercise going from scratch, really without any tool, any library, and we will go further from there with more resources and more links, copying, pasting, and understanding them. So if everyone is, you can also use your computer, personal computer. What we did on the cloud is just installing all necessary packages, so you don't have to. But then you copy the assignment, you create your scratch your new R file, and we can start. So the first exercise, for which we assign 10 minutes, it's really create a demographic table using only dplyr. It's really a classic table, as I already mentioned, and every submission has, I think, this table. The data should have, for simplicity, only age, gender, and treatment columns. Then I would include only the mean and standard deviation of age for the continuous data, and then a count and percentage for the gender gender part, gender character factors. Yeah. So we start with the countdown at 5. So at 2 minutes, I will add the how to make the data. I think it's very simple, but maybe I can help you to try. I did it. I did this exercise yesterday, so the solution could be very suboptimal. So be patient about the solution. It's really not a matter of what is how you get there. And in the meanwhile, if you have any doubts, anything, please write on the chat. Yeah, you have to create it. You can create it. Sorry, someone asked, I'm probably missing something obvious, but where is the data? So the data is, I mean, creating a fake data is part of the exercise. And I mean, all of this, it's really, you will see why we are doing this. It's really going from let's say, the simplest possible thing. So someone at a certain point decided, okay, I want to do it with only R, and started from this. So I have the demographic table, and I need to make it just from scratch. So if you are new to R completely, I think there are tons of things already. Also, we have done many workshops about how to get to use some of the tools that we will see afterwards. But in general, if you are completely new to R, I would suggest to start really from the basis, like a nice Coursera course. And then we have also, and it will be in the additional resources, it's also on the website, the Coursera that we made for clinical trials submissions in R with the NEST tools. For this specific exercise, I invite everyone to just use all the tools you have. Remember, you have, yeah, maybe Leena.
Yeah, I will send the link.
Of course, if you want just to see the solution, there is a solution already. On the website. The fundamental aspect here is that when you are creating something like this from scratch, you are not supposedly, and you're not alone, right? You have whole internet at your fingertips, so you can ask Google, now also ChatGPT, and I don't know, other artificial intelligence. It can help you for sure with the first part, with creating the fake data. Yeah, I mean, so there is a question about, again, about Shiny test. And I mean, if you go to the teal insights engineering, you will find the link on the website of our GitHub Insight Engineering project that has all our packages. And we'll find also teal there. And if you check there, you will find yeah, that is fine. Sorry, to finish up the question is you will find on the sorry, to respond again for Shiny tests true, you can check on the teal repository. There is the folder with tests, and it's all open source. You can you can see how they are achieved. And again, those tests are happening every day. We test everything every day for all together, and then all PRs are of course tested. We have all the CI/CD pipelines in place. Okay, 0. Yeah, also Pharmaverse, and I see Mike that is sending information. To start with our so solution. First of all, we need to create the data. So we create a data with a data frame, and so 100 patients, it's enough. So we create 100 IDs. They're simple numbers. Then age is just a sample between 18 years old and 80. Then gender is male, females. We will add more in the future, but this is just for simplicity. Treatment A and treatment B. And yeah, you can see the output is very simple. This is only the first 6 elements. As a code, again, this is what I find out to use, but you can create it. I'm sure you created different versions of this, and I think they are all fine. So first part, this one, which is of calculating the treatment counts. It was not requested, but I also did it. Then you just left join the treatment counts to the data, and you connect it with by treatment. So you create basically another column with the treatment label. Then you group by treatment label, which will be at the end what we want to have on the columns. Of course, the group by works on the rows, so at the end we need to transpose for easy review if needed. And this is a very simple summarize where I just get the mean and the standard deviation of the age in one character. And then I do the count and percentage of female males, and I drop the groups. Then you print it, and it should be something like this. I corrected it just to make it look nicer, but the output is the same. You have the continuous result and then the factors result here. So this is first exercise is to show you that you can do it. It's of course it's probably not production ready, but it's relatively simple, and it's I mean here it's 20 lines, but if you don't do the total count, the treatment counts is quite smaller. So here there is a bonus solution that is just the same, but with turn and R tables. So these are our packages, the core tabulation packages for making the same result as you saw before. So you load the mean, and then you create a basic table, which is only a shallow table with nothing in it. Then you split the columns by treatment. So you say, okay, I want to have treatment A, treatment B as a columns. So I divide the data by treatment, and then from turn this function analyze vars, I analyze gender and age. In turn, this function has all the summary functions that can calculate a different result for what is continuous and what is a factor. So we don't have to specify what is what. Lastly, we use this layout, which because it doesn't have the data yet, and we build the table by adding the data, so piping in the data. And the output, I'm sorry, I need to format it properly, but it's what we wanted from the other one, but a bit better, right? Because this is a bit more ready. You have numbers of elements for treatment B and A, and then you have female males, you have age, and so on. So now, exercise 2, it's an escalation of this. So how to create a demographic table like this. So this is taken from the FDA standard of safety tables and figures, an integrated guide. You can find it from the link that is on the slides. I don't think you can see the footers from this presentation, but it's on the website, so don't worry. Get there and have all the description of this table. But you will not need to consult this document, just see this and see how it seems a daunting task. But recently collaborated with Sanofi, BI, and Moderna to create this package, which is Cardinal. And I think this is very, very nice also because it has more than one solutions to the problem of creating a table like this. And so this exercise, it's just yeah. So first of all, you can just go to the website of Cardinal. You can find it is open source. Just write Cardinal package. You have all the links in the presentation. But if you write like just Cardinal package on Google, you will find it. And you will find that this is the table number 2. So check that out. And I already wrote here the data because I don't think it's very interesting to create this data set itself, because we have the cached data set with random series data, and you can just copy paste this, and we'll have the classical output of with a lot of columns, really a lot of columns.
Yeah,
So I would copy this code. Yeah, sorry, Leena.
Yeah, just to say, the random seeders data will have cache versions of typical ADAM data sets. So here we're calling ADSL and ADVS. So it's just dummy data, but it's very typical to what you would see in a regular ADAM data set.
Yeah, thank you. And we are also working with Admiral data for some of our tests. So it's just what we internally used at the beginning for testing and for producing the synthetic outputs for the TLG catalog that we will see afterwards. Yeah, so the output is, for example, I think this is ADSL, not this is ANL. And you can see the style identifier, unique subject identifier, country, and so on. So now, yeah, I think this is a spoiler. Now I give you 15 minutes, probably 10. Let's see how is the time to try to make the table. You can also use GTSummary if you feel better with it, or our table. So the first function here constructs it with our tables, and the second one constructs it with GTSummary. I should have installed Cardinal already there, so you can use it directly, this function, and just you have the table. And you can click here to see the source code of these 2 functions. if you go to the slides from the website, and you can try to do what I did for this exercise and what I invite you to do, which is create them from scratch by copying some of the important aspects from the cardinal functions. They are not complex table. Of course, there are more options in these functions. So like you can add like an overall total patients, total column, and but all the information it's is there. So you have all the functions. I mean, all the variables are defined, self-defined in that place. Yeah, it's the validation hub. Thank you, Mike. And it's a Fuse and R consortium. I'm not too involved with this, but I think it's quite interesting. There are like risk metrics. I think there is also packages somewhere. Just need to find the place where they are mentioned, the outcomes. Maybe we can add it to the...
Head to a break.
Yeah. So first of all, the first solution I propose you is our table solution. So again, this is this function just rewritten with less cardinal related function. This was the caveat. So first of all, the FS split is NA, it's coming from turn, so you need to load the library. This just transforms all NAs that are only character or all mixed behavior into our friendly NAs. And then here you can highlight the vars you're interested and their labels. Of course, you can just specify like one variable and omit the label, and it will just print the variable name value. And then you have again a basic table, show call count. So this is for showing the column count. So it will be the number of elements for each arm. And then split columns by, you divide your data set on the columns by the treatment. And then you add an overall level, and you do this with split functions, where you can modify your division between column. So in this case, you add the overall level, you call it a total population, and you put it as last. And then this very familiar analyze vars that is coming again from turn, where there are all the stats you would need. In particular, here we decided to, at least for cardinal's sake, so for rendering the FDA output, just to have these three, which are mean standard deviation, median range, and count and fraction. Count fraction, which is a kind of count. We don't specify any formats. We use the defaults, and then we don't remove the NAs. Append top left is more information on the top left corner that is needed for this table. Then you build the table with the layout, which is this, that hasn't seen yet the data, and you pipe in the data, which is the F, which is NL. Then you prune the table if you want. I think I just took it from Cardinal. So the output will be something like this. And it's I think it's very close to the one that we started with, which is that we wanted, which is sorry, too much, which was this, sorry, this one. Yeah, and next one very rapidly, the GT summary solution is on the exercises on the website, and you can go through it if you're GT summary, if you prefer GT summary. You see that you start the same, then here you need to select the bars, because then I said all, I selected all, so I just did press select. Yeah, that's because it's all printed. So there is a question about typo, so a character that is not printed, but that's an issue of because all here is in HTML, and I think there is an issue with Quarto and HTML, at least in this presentation, but it will not happen if you print it locally, I think. At least I didn't see this problem on Cardinal, so I think it should be fine. Yeah, so you have mean standard deviation, mean range. Yeah, it should be removed. Yeah, could be. Then all categorical, you have counts and percentage. Then you do a bit of label. You set the names of labels, and then missing if any, which is the same as false in an A remove. Then you have bold labels, a lot of decoration, modify header, just to have also the counts. Then you add overall with the total population, and then you have a statistical label. You change the labels. You add the footnote, modify the footnote, and then I think it's alignment. I'm not sure about that. And the result is something like this. So very similar. The output and the numbers should be the same. It could be interesting to see if they are the same. Remember that the analysis functions, they come from different sources, so they are the same. Kudos. Yeah, and that's all. You can during the break you can start with the. You can check this and then ask more questions if you want, or you just take a break, which is also very good. So the first line of the code, which one? Someone asks to show the first line of the code. You have everything on the website, so yeah.
Okay. So Davide and I will still be around if you have questions, but otherwise I recommend we break here for just less than 10 minutes, and then 1:30 we come back and head to part 2. So yeah, do take a break. It's very much well deserved, but Davide and I will be around if you have questions. Okay, see you in just less than 10 minutes.
So the question, if there is one continuous variable, keep true decimal place, this one?
Mm-hmm.
Yeah, I mean, it's you see here there is formats equal null. Now you just specify a function here, and then you can decide upon if it is one or the other. It's not super simple. You just need to. I mean, honestly, I would just say function x equal something. So this is I would just put the function x and then browser in and see how to do to deal with the X in a different way. In general, anyway, these formats are more like string based, so you can actually, so you can write so that what I told you is the complicated way. Okay, let's go with the simple way. Just take, for example, here, just copy this. I'm gonna do formats equal this, and then you do like XX. Sorry, I'm writing, you don't see, but you see the first mean and standard deviation equal XX, and you can change it. This is the standard. You would keep all the decimal values and just change. You do like XX point X and so on. And if you go, and I mean, you will probably make it wrong, and it will tell you it's wrong. Use this function that is a list of all the possible formats. Yeah. Maybe Lena, if you can find the formatters function that lists all the.
Yeah. Yes.
Q.
Okay. Should we maybe continue with the next part? I know there are some more questions being asked, but I think maybe in the interest of time, we can get to them towards the end. But if there's questions about the exercises, then yeah, myself and Davide will look over the chat for this. But yeah, do continue, Davide.
But don't worry, we have a lot of bonus, so that you can just look at by yourself if we don't have time. So for exercise 3, we thought about making something that looks complicated if you want to do it from scratch, but for which we have a handy way with our tools, and is an uncover efficiency table with a single visit and single endpoint. We decided to go with a single visit and single endpoint just for clarity, for compactness. There are also, and also for what is the bonus exercise afterwards. So for all of these, we could have started from scratch with making turn and everything we did. But then we said that this is not what what we do when we are asked for a new table. We just check the TLG catalog if we have already created one of these tables, or if it's similar to that table. If it's similar to one of the tables that we already have in the catalog, we just take that table and modify it accordingly, or copy and adapt some of the functionalities to get to that table that we need. The TLG catalog has more than 100, 200 tables and listings and figures, I think. So there is plenty to take inspiration from. So what I suggest is to pick at the solution. You can also try from scratch. I'm also happy with it. If you want to try from scratch, you just go from turn. But I recognize it could be an issue to find exact functions because there are a lot of functions. So I suggest everyone to just go to the TLG catalog and start it from there, trying to understand each function. So just a question mark function, you see the documentation, what it does, and you can also see the code. So there. So it's a tiny table. By the way, Leena reminded me that we also implemented a way to to try try things on the catalog, which is WebR. So if you click here, you can try to and use it. It's experimental use, but yeah, we'll avoid it installing all the packages now. But you can click on it and just work from there if you want a third way to work with it. You need to start, of course, from data setup. It's a very simple data setup, and you see the filtering of the visit and the param CD.
And do post in the chat if anyone tried with WebR directly on the TLG catalog. It would be nice to see.
Yeah. It's fairly recent. I think it has 3 months.
Yep.
This edition.
Yeah, very recent. We made a blog post in Pharmaverse blogs on WebR addition to TLG catalog. Great, good to hear, Tom.
Maybe Leena, you know how to answer this.
Yeah,
Can you please explain single endpoint?
Essentially, we are using questionnaire data, I think, for this, are we? Essentially, it's just a type of parameter in the Adam dataset, so it's completely made up. But if you would like to think of it as a type of param or param CD code in the Adam dataset, so just for a single variable at a single time point. But
Yeah, I think it's coded.
yeah, sorry.
I think it's coded as a param CD.
Yeah,
You can see
yeah.
here. So
As a param CD, yeah. And like we said, with these, the random CDIS data set, it's completely dummy Adam data. So it's possible that you find especially when you see things in the TLG catalog, like perhaps it doesn't make too much sense, the parameters or parameter codes, particularly for efficacy, but that's because it's entirely made up. And even for adverse events in this bit, it's not based on any true kind of AE event that you would see on a real clinical trial. So yeah.
but I must say that we added so many tables and things that it needed to be kind of realistic. So even if the values sometimes are like randomly picked from like a flat distribution or a Gaussian distribution, it's I mean, there are a lot of parameters, a lot of columns, and I think we have loaded like 10, 15 different cache dataset from the Adam repository. So I honestly would, I mean, I really think there is a lot of added value in this synthetic dataset.
Yeah, it's very easy to adapt to let's say if you were to copy and paste from the TLG catalog or any examples that we've shown you today, and then reinterpret this into your actual Adam data sets, I think is fairly easy to do so.
Yeah. Yeah, funny story that I was working on some Admiral data. Of course, it's all synthetic, so it's a matter of how it was built. But I remember that at the beginning there was like a lot of columns less in the Admiral data than OCG's data, because of course you didn't see so many. I mean, when it was designed at the beginning, it was not designed to be applied to so many tables. So we talked a bit with the Admiral team, and they helped us adding a couple of specifications, descriptions, and things. So yeah, the source is all synthetic. Yeah. No, we are not allowed to use actual clinical data, of course. It's all open source, and yeah, it's too risky. And yeah, I thought about this. I think it's a good question because at the beginning I was a bit, I say, I wanted to have more realistic things to work on, like mainly with distributions and these kind of things. But I mean, all the work you can do in that direction it has a limited added value at the end because there are, I mean, there are not so many edge cases that need modeling of the data. Augustine, what do you mean with multiple imputations?
Oh, I see. I would say multiple imputation would have to be as part of the processing steps. Perhaps there's another package that could handle this, but then you would be able to output this into efficacy tables. But I would say, yeah, that multiple imputation would have to be part of the pre-processing.
Good. So we finished the time, so let's talk about the solution. So the data setup, you already saw it. You just load in the cache of ADSL and the DQS, these Adam data. Then you ensure that the character variables are converted to factors, and empty strings and NAs are explicit. Which with this function, this is coming from turn, and then you filter for a visit and param CD. Yeah, and you add this. I don't remember what it is exactly. I think it's related to this table. And we will see it here. So of course this CHG. Do you remember what it stands for, Leena? Change, I guess.
Yeah, change.
Yeah, makes sense. Um, and okay, so first the basic table, we already saw it. Show column counts. It means that we will have the column counts on top. You see them here. If you can see them. Then you append the var labels. So you add the labels to param and for parameter. You add summary and cover in an adjusted way. So this is the first piece. So this is the first block. So if you only use this first block, you would have only this first part of the table. And this is a turn function. And as I said, you can check exactly how it works. And this is a regression test and cover regression test with as a reference group arm A. And you can see here when we split the columns, we specify the reference group. Then in this case, it's an adjusted, so you don't have any strata, any covariates. And you say confidence level of 0.95, and you get this first part with the 95% confidence interval and its p-value. Of course, it's very large, this p-value. You don't want this from your results, but it's because it's all random data. So again, summarize and cover, you can call it again to get the other part of the table. And in this case, you add the covariates of the baseline and the strata one that I'm not sure what it is in this synthetic data. Then you build everything and notice that there is an alt counts df, which is basically ensure everyone it happened with everyone that you need to add an alternative counts, so like the number of patients, and you just add it this way. So it's referring to the ADSL number of patients instead of the selected here. That's why the actually call counts is higher than only when you select a DQS, the single visit CD. Okay, good, good. Any question? If not, we can get to the bonus. I think we have time for one small bonus. Right, Leena?
Yep.
So
Maybe one bonus, and then we have to go to the next
yeah.
exercise. Okay.
So we could actually yeah, 10 minutes, fine. Leena, let's go.
Yep. Let's go.
Probably you can stop at 5 minutes. And again, sorry, but how TL would work with this table specifically. So let's try to build a TL interactive table now. So this table specifically, and of course, again, you don't have to reinvent anything because it's all in the TLG catalog. And it's referring to the TL modus clinical package, which collects all these TL functions that are specific to they are specific to this to specific tables. Let's say. In this case, you can find it here. So after the table that we just discussed, and we also implemented recently Shiny Live. So if some people want to try, maybe I will try. I think I never actually tried. Leena, did you try ever this Shiny Live?
No, no, I haven't.
So I will not. I mean, again, if you 10 minutes are way too little for going from scratch, so I would just copy and play with it. So copy the result from here and play a bit with it. So you start understanding some functions like we have till data. So I mean, for me, I start to think how I can put my data in, and these kind of things. So you can't execute it in the TLG catalog. I don't know. I think this is all based on GitHub. So if we all click together, could be an issue. So maybe it is crashing something. I hope not. But if it works on Posit Cloud, I think it's good. That's the important thing, that you're able to host your own Deal Modus Clinical code. And yeah. So as usual, I show you the first part, that is the part about the data initialization. So with TL, you need to be a bit more cautious with what you put where. So here in particular, you need to add your data set into a block code that is within this TL data object. And then you need to specify a couple of things, like data names Unjoin keys. Oh, wait. We have something which is still loading, so I think it still needs a bit of a moment because we don't see the table. Anyway, you have a couple of configuration for reusable configuration for modules. In this case, you have like the ref, this the default ref, and actarm cd, which is I think it's, similar. Okay, okay, Shiny Live works. Takes 5 to 7 minutes. But I think, I mean, we will see where you can find all of these in a nicer way and in a faster way, let's say, because this is all hosted there. Anyway, here you can find one of the motivation that made us pick this table in particular, that you can just change the filter live. And you say, okay, I want to introduce two visits, and it just does it slowly. I'm sure you already played with all of this in your local. Anyway, you see that there is also week 2. Then you can of course change a couple of things like, I don't know, age. You can add a filter and you reduce the number of you filter out some variables. Okay. I selected age units, which is not what I wanted.
Yeah.
Okay, I think we are almost at the end of the time allowed for this exercise, and you can see that to set up the app, you need to initialize it with the data and then insert the modules. And these modules are already defined in Intel Modules Clinical, so you can just check the code there. And it's highly structured our tables and the third. And you see that you just play with this, like this is the label of the module, so of this app, and then you see that you can add one of these selection choices for a visit. Personalize visit here with a selected value, and it's the same for all these variables. Okay, we got the age. So here it should be a plot. Anyway, you can select, change this, and then everything else will change. But I would close this now because I feel like it's taking a toll to my computer. It's just slow. I think it's just the backend. It's just the GitHub. I feel. Yeah, anyway, you see the plot. You can also, the histogram, you can also slide and select things. And now it's updating here. So, okay, good. Okay, we had even more time, but I think it's fine. I hope everyone understood. So next, we have another bonus in which you can use the data from exercise 1 to do it. So to create a demographic table. And we added it to the exercises. So if you go on the website, you can see this. this solution. I think we don't have time to cover it exactly. It's quite straightforward. I just copied the demographic table output for TL and added my data. So another exercise, the Kaplan-Meier plot. So of course, everyone I think was interested, at least I hope it was interested about plots and plotting. And we also have a lot of figures in TG catalog. So I invite everyone to check there and try how it works.
So after this exercise, we have exercise 5, the TL gallery, which I really would like to show people as an alternative to using just the TLG catalog. And then we can head to takeaway. So maybe if we spend about 5 minutes creating a KM plot.
I think we can probably just describe it if it's
Mhm.
okay for everyone.
Mhm.
And you can play with it from the TLG catalog after the workshop. So the data setup is just filtering and then selecting a list of variables, descriptor, and then you just call the turn code for this, and you get this kind of plot. There is the Cox regression box is this one that you can add if you want. Of course, you need to have all the options in ANL, so your dataset needs to have all the needed characteristics. And in particular, these variables that I showed you before in the data setup are the main drivers. So like is event is the yes no, so true false that makes the regression that is the main test for the Cox regression. Yeah, and you can see that also arm is the three arms, yeah, that you have here. Okay, this is without comparative statistic, as you see. Yeah, and there is no that part. There's not this one. Again, you can try it with WebR, and you can also use TLAP, Shiny Live, which works, I think, very well, honestly. And then it's very similar also for TL here. You have first the data settings, then you set up the app with the modules, right? This is the module that is TMGKM, so the Kaplan-Meier graph, and you specify like which choices you want to be able to select and so on. Yeah. Sorry for speeding this one, but I think it's quite self-explanatory. So lastly, we have the TL gallery that I think it's very nice to understand how powerful TL can be. So this is a bit of the end of our journey. So we started from creating a table from scratch, and we ended up in having an interactive hosted server of apps. And again, if you click on the TL gallery link, you can also search for it on Google. It's all open source. You can find different possibilities. We can click, I would suggest to click on efficacy because we talked about efficacy so far. I think this is hosted on Shiny Apps. So I think this is some server. So it should be faster. And it is faster. So it's already there. And you say, for example, demographic table, the one we're working on, here it is. And now we can more rapidly select a different range for age, for example. So let's suppose that the FDA asks, okay, but what about people between 20 and 15? No, this is impossible. 25, 35, let's say. and then you can just peek at it, answer maybe with this say an informal response. Of course, you have also exports here, and on the in the back end is the same table, same everything is just filtered output. So yeah, you could give this back, for example, as probably an informal answer. And then if they want a complete answer, you should probably go back to the original data and filter it out at the source. We have a lot of other things here, so I invite you to play with all of this. It's a bit stretched out, but again, is a syntactic data set. And we have also the Kaplan-Meier plot we talked about. Of course, when you save it, when you export it, you can save it with a different size, and you can also report stuff. So you like add to the report, and there you can also specify other exports like HTML, PDF, Word, these kind of things. Leena, do you want to add something?
Just to say actually how powerful TL modules are. It's very nice when you're creating static TLGs. And for example, if your stakeholder wants to see different variations of a TLG without having to recreate again, it's very useful to explore and have a taste of the TL kind of framework alongside your static components for deliverables. Most often I find TL apps very useful for stakeholders and answering quick on-the-fly questions, for example, in meetings, or if there are questions in particular about anything to do with any of the modules. I find it most useful. Another cool feature, maybe we won't go too much into it, is the reporter module, which is really nice if you want to kind of save the progress you've made already in your app, and add this as a PDF kind of output. Of course, if you are going to share it with stakeholders, you have to add a disclaimer that it's produced via exploratory means. But I would say this feature has been a game changer for TL because yeah, it helps you save that kind of state that you've created in TL and then recreate it if you wanted to as a static TLG, or share with people who may not have access to the app, etc. So yeah, it's a very powerful thing, and I think the journey we've shown today as well, just from going from a very generic basic demographic table to all of these wonderful functionalities we have, yeah, has really shown the progression that we've made within the Nest team.
Yeah, like here you see a glimpse of a report for the MM RM table output that I randomly picked and calculated, and you have also the specific of correlation structure, adjustment method, and also some the filter state. I remember the discussion we said about cherry picking biases. Here you have everything stated, and yeah. So I think it's that's all for the technical aspects of the workshop. Now Leena will give you a final perspective on what do we expect from the future submissions and future regulatory submissions. Yeah.
Yeah, thank you, Davide. And well done, everyone, for sticking through it and having a go at the exercises. Like we said, the materials will be on the webpage that was sent around in the chat room. I'm sure we can make it available afterwards. So yeah, if you wanted to go back or look at anything in particular, then it's all there for you. Right, okay, so we'll round up today's workshop with a few conclusions. So what do we expect? So in the short term, there are some considerations to be made when you're kind of stepping into this new world of possibilities when using R and open source packages. One of which we are kind of in the midst of, I would say, at Roche, is that the migration process requires some initial investment in terms of time, resourcing, and training. A lot of effort was made not only from kind of a top-down approach, but on the ground as well, to really influence and change the way that we work. And having those people around who are contributing to the packages and developing, not only that, but people who are actually laying the foundation for the ways in which we work is really important and a great investment to have in any case when you're shifting the dynamic. Existing workflows and scripts, of course, need to be rewritten and adapted. This again is a lot of overhead, I would say, but it's well worth it. And it just provides you with that flexibility in terms of the way that you work. And in the longer term, Davide, if you go ahead, click to the next.
Wrong clicking.
Yep. No problem. Yeah, but there are some wonderful long-term benefits from doing this. So we've mentioned them previously a lot earlier in our workshop, where we mentioned the cost benefits and increased flexibility, access to a broader talent pool, and also staying ahead in terms of technological advancements. But I really wanted to highlight here that it is the open source nature, the transparency, and reproducibility of our results, which we are finding to have an excellent benefit in the longer term. And efforts such as the one that Davide walked through, the cardinal effort where we're standardizing TLGs across company, is really just showcasing the possibilities and the longer term gains from this shift. And of course, if regulatory bodies continue to increase the acceptance of R, then the case for migration becomes even stronger. So like I mentioned before, to see more continued success when we submit end-to-end in R is just a great benefit for us as an industry in general. And of course, all of these use cases for R then becomes even stronger. Davide, any other comments from you or your?
I think you said everything. Of course, we started all our journey with three things that were reproducibility and validation. And then we also talked about open sourcing and interactivity. And all of these three things we strongly believe will be the future of submissions. And we can see that we are getting very close to that. And as Leena said, we're already long-term benefits coming our way, and of course this becoming a global effort. I say global, but let's say a community effort means that at the end things, all code and everything is tested much more. There are more people ask for bug fixes and check the code, collaborate with the code, and at the end you have a code base that is getting things forward faster, and this is all what matters to get clinical trials faster to the submission protocol, and to get faster new methods and apply faster new solutions. Yeah.
Great. Okay.
So, Leena, sorry.
Yep. That's fine. Okay, so where can you find out some more? We gave you just a bit of a flavor to everything that we've been doing in Nest and how we experience things at Roche. So if you want to find out more, of course, you can see all of our source code as it's open source. We've mentioned so many times, I've lost count, on our GitHub organization there. It's linked. Then Nest is also part of Pharmaverse. So we frequently also blog on the Pharmaverse blog site as well. But if you do want to find out more about Nest, then yeah, feel free to check out Pharmaverse website as well. A few of the things that we've mentioned today during the hands-on exercises, which is the Cardinal project. So I thoroughly recommend you to check it out and see side by side kind of the different ways in which we create TLGs and the standardization piece across company. Also our TLG catalog, which we've seen in depth today, which is great. And also the TL gallery. Again, both of those products, we really just scratched the surface of today. There's so many more possibilities and types of outputs for regulatory purposes, and also very much more on the exploratory side, especially for TL, that you can have a look at and see. Okay, and something we mentioned right at the beginning off of someone's question, which was how to learn more about R in general and in the purpose of our industry as well. A group of data scientists at Roche Genentech created a Coursera course, which is open to everyone. I think it launched a few months ago. And we developed a series of trainings really going through the journey of statistical kind of programming and for stats in our industry. So there's a chance here to explore how to create ADAM datasets using Admiral, static TLGs using Nest, and interactive data displays using Teal, as well as that. So it's very much more in depth than what we had covered today, especially for the TLG portion, but it gives you the context around the reasons for doing this as well. So more context sharing, I would say, and a lot more hands-on guided practices and deep dives into the workflow as well. So I'd really recommend that you sign up for the Coursera course. I also would say that it's a great kind of springboard, I would say, for new hires or new joiners in your company to say that they've already done a course like this one and already have learned certain aspects about kind of our workflow and our processes and the open source packages around them is really good. So definitely within our organization, we recommend this new joiners having already did this prior to joining or as they start off their careers to do this course. So do check it out. Okay, and I think this is the final slide, if I'm not mistaken. Is on collaboration and how you can get involved. So if you do notice a bug or you have any ideas for enhancements, then please do get in touch. Next slide, Davide. Maybe we're lagging on this. Yeah, please do get in touch. We're always open for collaboration and we really do encourage us to check out our code base and contribute with us, whether you're an individual or a company. We really like to hear from you and develop this together. We're also on Slack. You can find us on Pharmaverse packages if you have a general question or you just want to reach out to us or hear about latest and greatest developments within the Nest team. And yeah, if you're interested in industry collaboration, please do get in touch with me. I've left my email there. Or if you have any questions about the workshop or anything in general Nest related, then I'm happy to answer. Or yeah, like I said, for industry collaborations, please do get in contact via email. Okay.
Yeah, if you want also to submit a feedback, would be great. Not mandatory at all.
No, yeah, not mandatory, but really to help us out here, it would be we'll be grateful if you fill out our feedback form. It'll only take 5 minutes, so maybe while you're getting a tea or a coffee, just have this on and fill it out for us. It will help improve our sessions in the future, and then we'll understand as well how you found it, and yeah, what we need to improve or what we can potentially add in future workshops. So please do fill it out. Okay.
Yeah. I think
Good.
now, if there are questions, we have like 5 minutes.
Yes, 5 minutes for questions if anyone has. Yeah, feel free to come off mute and ask, or post in the chat or the Q&A box. There was one question about introduction to or like basics of R programming. I did put a link for a blog post on how to learn R as a SaaS user. I mean, there's so many other resources also available, LinkedIn Learnings, for example, or even R, like the cookbooks, and yeah, there's so much out there already which is open for everyone. So I'd highly recommend that, and maybe we can add something also to our website, Davide, as a pointer for new users.
Yep. I think it's a good getaway for this information. Yeah, and there's also a clear pro of migration into R. We already mentioned it at the beginning, that you have so much resources to learn. There's so much open source information and courses.